Yeah, so our next speaker is Tanmay Bharat from the LMB, talking about combining mass spectroscopy with cryo-EM.
Thank you very much. Thank you also for the organizers for inviting me. I see lots of friendly faces in the audience. Very excited to be here. To tell you about how we are thinking about combining mass spectrometry with cryo-EM. Let me just show you this nice image of a cell. So this is the smallest known free-living cell, Nitrosopimulus maritimus. And using electron tomography, we can produce beautiful pictures like these, where we can see, the internal components of the cell, the nuclear material, ribosomes, and we can image down to the molecular level. So this cell is surrounded by a surface layer. And the surface layer, just to give you an idea, there are these hairs on the S layer, which are individual glycans. So you can get very high resolution pictures with electron tomography. You can even push the resolution even higher by subtomogram averaging, and you can solve, so we've solved the structure of the S layer. And you can get, structures where you can directly do atomic model building. So cryo-EM, this is not news to this audience, provides us with high spatial resolution pictures, which we can use to gain really high resolution information of our specimen. I know many people in the audience are working on in situ structural biology, so this is not really new to them, but this is our kind of thing. So this is our contribution. So cryo-EM can now be pushed to tissues and organisms. So this is zebrafish embryos that are infected with mycobacteria that we have imaged with cryogenic volume SEM and also with tomography. Now, there's a problem with this kind of images. So as you go higher and higher in complexity, it's very hard to know what you're looking at. So what we end up with in electron microscopy is basically black and white pictures where we don't know what we are looking at. And this is a very difficult problem to solve. How do you know what you're looking at? So this is an image of infected zebrafish with mycobacteria. You see the bacterial cells, you see the surrounding host tissue, and you can even label it with prior knowledge that, oh, maybe this is a lipid droplet, but you don't know for sure. So this is all based on people's knowledge, but no direct evidence. So what are the current available methods to ID things in images? So you can either, as I said, solve the structure of your target. So this is now going back to the S layer. So this is from a different bacterium. We, to know what we are looking at, we had to use X-ray crystallography, cryo-electron microscopy of reconstituted systems, and electron tomography subtomogram averaging to finally figure out where this molecule is, what it's doing, how it's assembled. Another alternative approach, which many people use, is template matching. But then, of course, you need prior knowledge about your molecular target, and also template matching also has limitations. So basically requires prior knowledge of the molecular target. Another approach is to provide a fluorescent label to your specimen. So you can label your molecule of interest with a GFP or some other fluorescent staining. And this is something we've done here on this S layer to study how the S layer biogenesis occurs, which gives us very powerful information. But of course, it's still limited because you can only label a finite number of things in your molecule, in your specimen. So this leads to the topic of this talk, which is combining electron microscopy with mass spectrometry. So mass spectrometry is a very powerful technique because it allows us to ID molecules directly without tagging them. And we started working on this, combining these two techniques many years ago when I was still in Oxford. So this is again the same S layer that I showed you in the previous slide. To understand how this S layer is arranged in the cell, we can of course do subtomogram averaging, but we can also do native mass spectrometry. This is a technique developed together with Carl Robinson, where we can, where they could directly inject these S layers bound to the membrane into the gas phase and do native mass spectrometry, where we could find the dimer, trimer of the S layer, which are which we could map back to the dimeric and trimeric linkages found in the lattice. This was a very informative project for us to understand that these two techniques can be combined. Also, so this is just another way of telling the same thing. So here we have combined cryo-electron tomography to understand how this S-layer molecule is assembled on the cell. And we reconstituted the S-layer protein together with this polysaccharide to get hints of how this molecule could be arranged on the cell surface. Despite all this structural information, there were gaps in our knowledge, which could only be plugged by mass spectrometry. And in the case of mass spectrometry, we took these complexes and we did native MS, which told us the stoichiometry of the complex, which allowed us to count, in this case, how many sugar moieties were bound on the surface of this cell. And it was actually 42 hexose moieties, so we could do single hexose counting by combining cryo-electron tomography with mass spectrometry. But this is something many people do. They combine cryo-EM with mass spectrometry in separate experiments. So you do, you take your specimen, you do one experiment where you do cryo-EM or cryo-electron tomography, and then you take the same specimen and you send it for mass spectrometry. So this is an indirect combination of the two techniques. What we wanted to do was to combine the two techniques more directly and apply them on the exact same specimen. So still back from our Oxford days. So this is a technique that we developed together with Stefan Rauschenbach in Oxford. So our role was the cryo-EM bit, and their role was taking these purified specimens and or not purified specimens and injecting them into the gas phase. And then they could be mass selected, and you could deposit these specimens directly on an electron microscopy grid, which at that time was at room temperature, and then they could plunge freeze it and perform cryo-electron microscopy imaging. So this first demonstration, here we got very nice class averages of this beta-gal sample, but the resolution was limited due to dehydration effects in the gas phase. Next, this system was developed further, and now deposition could be done directly in grids that were cryo-cooled. And then redeposition of the ice led to samples that resembled more like a cryo-EM sample you would see. So this is a plunge freezing control. This is the sample that has been deposited on a cold grid and then slightly rehydrated by ice deposition. It looks like that. And this led to high resolution structures of this molecule, which admittedly differed from the solution structure that you would get from standard cryo-EM, but is a powerful demonstration how we can combine mass spectrometry and electron microscopy directly on the same specimen. So it's just the introduction. So now the main part of the talk, can we do the opposite? So this is where we wanted to develop further. Can we do cryo-EM first and do mass spectrometry afterwards, and of course on the same specimen, so that we can figure out what exactly we were looking at when we were doing the cryo-electron microscopy. And this is where our laboratory is working. We have made the first start, and hopefully we will do more in the future. But so the way we are doing it is we are using secondary ion mass spectrometry using a focused ion beam. So we have a focused ion beam instrument which shoots focused ion beam directly on the specimen. The beam is rastered across the specimen in a pixel by pixel manner. And in every pixel, there's an extraction where the ions are extracted into a mass spectrometer, which leads to a pixel by pixel mass spectrum. And this pixel by pixel mass spectrum can then be converted into an image, depending on whichever ion you're interested in. So in theory, you have a mass spectrum at every pixel, but when I show the images, they will be just focusing on a single ion from that spectrum. And the hardware is not terribly complicated. I mean, yeah, it's, let me just rephrase that. It's standard setup in many material science laboratories. So it's not like we are reinventing the wheel here. What we are doing here is in a focused ion beam instrument that is equipped for cryo work for biological specimens. So this is what it looks like. We just have an extra thing in our focused ion beam instrument, and it's a standard. This is a Zeiss cross beam, but I think it can be in any commercial instrument available. So this is also my role here to popularize this technique. So when you buy your next FIB SEM, please consider just asking your vendors to put a SIMS device in there. So this is the SEM. There's a focused ion beam in there. You also have the SIMS extraction optics, which remove the ions directly from the specimen that have come off due to the action of the focused ion beam. So we have this nice setup. Can be applied to a real world problem. So this is where we, just to show you that this is actually a useful technique. So one of our collaborators in Cambridge, Kiran Patel, they were studying the accumulation of the so-called forever chemicals. So now this is all in the news. And actually when this study was published, we had a lot of BBC and other exposure. So they found that these forever chemicals were accumulated, bioaccumulated inside human gut bacteria. And they had these nice curves to show it. However, the reviewers of the paper didn't believe that these molecules were going inside the cell. So this is where we came in. And this is what one of these forever chemicals looks like. So this PFNA, so PFAS, whatever. So if someone is working on that. So it has all these different flowing atoms, which is very good for us because we can detect elemental ions very well in our mass spectrum. We also detect small molecular ions, but I will comment on that later. So for us, this was a very trivial experiment. So the experiment we did is the following. We exposed human gut bacteria to these forever chemicals, and we simply measured with our focused ion beam FIB SIMS whether this molecule is present in the cell. So we found, so this is a cell, this is a secondary electron FIB image. And when you do the mass spectrum, we see a strong peak for flowing inside this bacterial cell. You can see it, and this is a phosphate peak. But when we didn't expose the bacteria to these forever chemicals, we didn't see a peak for the flowing. So this was a very trivial experiment for us, but it was very important for the people who were doing this study. And also it convinced the reviewers that the chemical was actually entering the cell and not cell surface associated. Another nice thing about this technique is that three-dimensional imaging is possible because you're directly ablating the materials and you can just mill through the entire cell. So this is this example is showing that. So you raster the beam. Every time you raster the beam across the cell, material is being removed, and those ions you are actually collecting and measuring a mass spectrum, and then you can mill through the entire cell, and you can see the flowing signal is coming from inside the cell. If anyone has any comments on that, I can explain a bit later for the really technically oriented people, but you can clearly see the signal is coming from the cell. So FIB SIMS is a very useful technique to study uptake of drug molecules, uptake of different things, and to basically track where your molecules are. You can also do clever experiments where you can, so this is an experiment where we introduce C13 labeled starch and measured the uptake by human gut bacteria. And you can see, so this is Bacteroides theta iota micron. You can see when we put C13 starch, there is a C13N signal coming from the cells, which doesn't appear in the control where you don't put the C13N. So you can do these kinds of experiments. So I hope I've convinced you that FIB SIMS is a useful technique that can help us map molecules inside cells and give us some important biological information that you can't really get easily from any other technique. So now can we combine it with cryo-EM? The answer is yes. So this is how we've done it. So how we are doing our experiments is we do the cryo-electron microscopy once, or tomography, whatever you want. And as soon as you've done your cryo-electron microscopy imaging, you can take the sample, put it into the FIB SIMS, and then do secondary ion mass spectrometry, which gives you multidimensional information with mass spectra at every pixel. And then we can overlay this information with cryo-EM, which gives us not only the higher resolution provided by the cryo-EM, high spatial resolution provided by the cryo-EM, but also gives us information in the chemical dimension, allowing us to ID what we have actually imaged with our high resolution imaging with cryo-EM. So, sounds very nice, but can we prove this? So this is a proof of concept experiment, again using our favorite S layer. So this is the Colobacter crescentus cells, which are covered by this. Oh, sorry, I should explain what an S layer is. An S layer is a paracrystalline array, approximately two-dimensional, which coats entire bacteria and archaea, and they are ubiquitous in prokaryotes, nearly ubiquitous in prokaryotes. So here we have used the S layer to tag these cells with a gold nanoparticle, which is 5 nanometers in diameter. And this here, you can see the cryo-electron microscopy image, and you can see that this cell has been successfully coated with these gold nanoparticles because it's surrounded by this S layer. And this is the cryo-EM image. And then when we go to the mass spectrometer and do our Fib-Sims imaging, we can see a very clear signal for gold. And then we can take this SIMS image and overlay it with the cryo-EM picture and basically map chemistry back into the cryo-EM image. So the advantage of the technique is that you don't just get the signal from what you tag, you get signals from everything that's in the sample. So here, these are untagged bacteria. And these bacteria, these are same Colobacter crescentus bacteria. They have this cell envelope, and they have these storage granules, which I'm going to focus in this next experiment. So here, we've tracked these different ions. So this is the sodium image, this is the magnesium image, and this is the potassium image. And you can see that magnesium and potassium are highly concentrated in these storage granules, while sodium is present in the entirety of the cell. So now you can imagine that this kind of technique can be very powerful in mapping these kinds of things. Also, I should say, when we are running the mass spectrometer in positive mode, H3O plus is our strongest peak, which will also allow us to measure pH of different parts of our specimen. Okay, so we can combine our mass spectrometry with the cryo-EM. Can we go even further? Because cryo-EM, you can do many things before doing your cryo-EM. So now this is an experiment which is, which was a heroic experiment because it involved many different things. So here we have taken two different cells, the same cell, it's a Colobacter crescentus cell, which we have applied two different labels. So one set of cells are labeled with the GFP, shown in green, and the other set of cells are labeled with MRFP, as well as a gold nanoparticle. And here in this cryo-light microscopy image, you can see that we can clearly distinguish between the two cells. So this is the MRFP labeled cell, this is the GFP labeled cell. So now we can do our cryo-light microscopy, then walk to the cryo-EM, then do cryo-EM imaging. And here you will see the same two cells. The cell on the left, which had MRFP and gold, is clearly labeled also with the gold nanoparticles. But the GFP cells have no gold label, and you can just see a cell that's unlabeled in the electron microscopy image. So this would be cryo-clem. And then we can take our specimen and then go to the Fib-Sims. And then in addition, do this mass spectrometry imaging. So you can see a clear gold signal from the magenta cell on the left, whereas there is a phosphate signal from both of the cells, as you would expect, because all cells contain phosphate, at least all the cells we've looked at. I don't know if there's some weird life form out there. So now combining all of it together, we can overlay the cryo-EM with the light microscopy, with the Fib-Sims, to get very powerful multidimensional information that you can't get easily with any other techniques. So now, so this is the full workflow of the whole we have developed. So now can we, so I think this is the last experiment I'm going to tell you about. So can we use this technique for discovery science? Can we discover some new things that cannot be discovered without using this technique? So here again, using this fluorinated molecule. So this is bisphenol AF, which is used in the plastics industry. And it's been, I believe it's been banned in the EU because of its disruptive effects. It's an endocrine disruptor. It's an environmental contaminant. So our collaborators, Kiran Patel's lab, they were studying, again, uptake of these molecules in human gut bacteria. And this is the experiment, and also environmental bacteria. And this is an bioaccumulation analysis. So you expose this pollutant to environmental bacteria, and then you can see accumulation of this molecule in the bacteria. It's not found in the supernatant, under different conditions, either in media or in PBS. And so this is a true chemical effect, because if you look at a control, there is no bioaccumulation. So this unrelated control molecule in the same assay has no bioaccumulation observed inside these cells. Another interesting observation, so this is a proteomics, so triplicate proteomics experiment, is that when this molecule, so bisphenol AF or BPAF, is exposed to these bacteria, they upregulate these antibiotic efflux pumps, presumably to remove this molecule from their cytosol. And now this led us to a conundrum. So if the efflux pumps are being upregulated, then how do we explain the accumulation of these molecules within the cytosol? So this is something we wanted to answer. And we thought our cryo-EM Fib-Sims method could be ideal to understand this. So this is what the cells that have been exposed to this pollutant look like. So these are the same environmental bacteria, Colobacter crucentus, which you can find everywhere. You know, if you go to the swimming pool, you smell something weird, it's probably Colobacter crucentus. It's a freshwater bacterium. You can see it's everywhere. So yeah, so when we imaged the cells exposed to this pollutant, we saw a larger than usual number of these storage granules, which we thought was a bit suspicious. So then we took these cells, and after doing the cryo-EM, we did the Fib-Sims again. And here we saw a very strong fluorine signal coming from these storage granules, which differed from the phosphate signal, which was coming from the entirety of the cell, as would be expected. So, and then if we do a high resolution, high magnification overlay, we can see that the fluorine signal directly originates from the storage granules, telling us that the the fluorine and the BPAF is going inside the storage granules, which is unavailable for efflux by these efflux pumps. So even if the bacteria upregulates its efflux pumps, it doesn't really help, because the molecule is unavailable for export, because it's now part of this phase-separated storage granule. So now I hope I convinced you that this mechanistic insight would not have been possible if we didn't have either of the techniques available, the cryo-EM or the Fib-Sims. So this is very important, that this technique can enable discovering new mechanistic biology. So just to show that it's not just a single image, so now there's more images of the same specimen sample that's been exposed to this pollutant, always gives signals for the fluorine from these storage granules, and here shown in the overlay. And I will just also show the controls here. So there's a control of an unexposed specimen. You can see if we look in the fluorine channel, we see background signal. And although I didn't mention the counts here are 1,000-fold less than in the previous slide. So yeah, so this is really background. And if you look at the phosphate channel, you see signal coming from the cells, as you would expect. As a final thing for this experiment, I would just like to say that we quantified the sizes of the storage granules, and we found a highly significant difference between the storage granule size when the environmental pollutant had the bacteria had been exposed to the pollutant compared to the unexposed bacteria. And we measured thousands of storage granules. So that's the last experiment I want to present, but I just want to kind of reiterate the message a little bit, that these the problem of identification is not going to go away, and it's going to get even more difficult as the field moves into multicellular specimens and tissues. So this is just example from our laboratory. So in our laboratory, I didn't present this today, we study bacterial biofilms. And we are most interested in the extracellular matrix that is rich in filamentous molecules. So we have spent the last 8 to 9 years solving structures of these filamentous molecules. And we've solved a lot of structures of different molecules from this bacterium, Pseudomonas aeruginosa. And I'm sad to say that knowing these structures despite knowing these structures, it's very difficult for us to identify these molecules within these kinds of images. The problem is this. The problem is that the molecules, they have a tendency, they could aggregate within this multicellular specimen, and also they could form a complex with other molecules. And also they might be in a different conformation, which might confound your analysis. And also should be said, which is something Bogdan Todor will talk about tomorrow, there's a crowding problem. So often your molecule of interest is in a very crowded environment, which sometimes confounds the analysis, template matching analysis that many people are employing in the field. Why I wanted to show this, that these techniques are also applicable completely to these multicellular specimens. So this is a multicellular Pseudomonas aeruginosa bacteria, where we made a lamella. And this lamella, after cryo-EM image or cryo-electron tomography imaging, can still be imaged with a FIB-SIMS, and we can do an overlay. And this is something hopefully we can extend in the future. I know that not everyone works on bacteria, so I just wanted to put one eukaryotic example. So these are yeast cells that we've plunge frozen on the grid. And here you can see clear differences in ionic concentrations or ionic signal coming from different compartments. So we believe this is the nucleus, which seems to be rich in, it could be a vacuole, so I'm not a yeast expert, but there's a higher magnesium signal coming from this compartment compared to the rest of the cytosol, where you see a high signal from potassium. I should also say that we have detected mainly elemental ions, and we can also detect small molecular ions like phosphate, Cn- in the negative mode, and we can also detect single amino acids and single nucleotides depending on the concentration. However, we would like to extend this method in the future to detect larger secondary ions, where we can potentially detect lipid molecules, peptides, which will be of incredible about other things, but just want to highlight